From Microbe TV, this is TWiP, This Week in Parasitism, episode 154, recorded on June 26th, 2018. I'm Vincent Racaniello. And joining me today, Daniel Griffin.
Hello, everybody.
Gorgeous day, isn't it?
Oh, it's spectacular.
Really nice. I was in D.C. yesterday. It was also quite nice. Lovely,
Oh, that's
lovely weather. And we have another Dixon-less TWiP.
Dixon-free.
Dixon-free. That's right. Dixon is traveling. He's been traveling since our last one, and he's still traveling. I'm going to meet him on Thursday in Austin, Texas for TWiV 500.
Well, that's exciting stuff. That is great.
Yeah, it'll be fun. Someday we'll hit TWiP 500, right?
I imagine so. Hopefully all three of us will still be around and doing well.
Well, you know, if we do 2 a month, that's 25 a year. It's going to take another 10 years.
That's right.
Who knows?
Well, actually, yeah, I guess I'm trying to think. So we only have to do about 50 more. So if we do...
Well, to make 500.
Yeah, that's true.
50 would hit 200. So 2 years would hit 200, but then we need another 6 years.
Intimidating number.
It is. It's just far off. Yeah. So I don't think we should worry about it. Just do each one at a time, right?
Exactly.
We have before we get to our case, we have a follow-up from Cullen, who writes... I just finished listening to TWiP 153. Now, Cullen won the book on 153. I almost didn't submit a guess for the last case study because I thought I was going to be late, but I'm glad I did now. I was also pleased to hear someone else follow up on the term PEEPs. Yes, PEEPs is short for people. I'm glad you guys got a kick out of that. I was not at ASM Micro, but my boss was there looking for a new sequencer and Malditoff. I always enjoy listening to your podcast. Thank you for everything you do on TWiM, TWiP, and TWiV and Immune. I wish I could have seen you in action at ASM Micro, but I guess that means I'll have to catch a live recording later. Cullen. Thank you, Cullen.
You know, I was thinking as you were reading that, my initial thought was, wow, every time someone accepts a book, they should have a little acceptance paragraph because they do, right? They have to email you back to give you their address. So they could throw a little acceptance paragraph in like that, which is nice.
Many of them do, and I don't typically read them because it's usually, wow, I'm excited to win the book. Here's my address. But this one had a little other extensive stuff here, so I thought I would read it. Then we have one from Lucian. Why don't you take that one, Daniel?
Certainly. Lucian writes, Dear Twipanosomes, this is a minor comment, but I think an important one. I think it was when Daniel was speaking about soldiers in the Middle East getting leishmaniasis, where he used the past tense when we had soldiers in the Middle East. I am paraphrasing. I just want to remind folks that the U.S. still has regular troops in Iraq, Syria, and Afghanistan. Last year, there were 26,000 troops deployed to the three countries, though the Pentagon announced a drawdown in Iraq starting in February of this year. I am not sure of the total now. The wars in the Middle East and Central Asia never truly ended. And I'll actually comment about that. And thank you for bringing that up. I actually think that's an important point. A lot of people forget, right, that the U.S. is seemingly endlessly at war. And what I was describing, I feel like Dixon sort of apologizing for myself, but back in the early 2000s, so I believe it was about 2004, there were a number of cases which they sort of realized were cases of visceral leishmaniasis in soldiers returning from what was referred to at the time as Operation Enduring Freedom, right? Mm-hmm. And so... yeah, that's an interesting issue. I do not know currently what the rate of visceralization is of the leishmania, or whether it was something specific about what was going on this. But that was the first description, and once something becomes commonplace, it doesn't get reported quite as much, which is sort of unfortunate. So I'm not sure right now, but yeah, that's what I was talking about. And thanks for the sort of pointing that out, Lucian.
All right. And now on to our case.
case from TWiP 153. And this was a brief one, and I'm looking forward to people's comments. But let me remind those who are tuning back in, and let me present this for those who are tuning in for the first time. We presented a man that I had seen I say in clinic or my outpatient office. I'm not sure what the distinction is, but my private practice out here on Long Island. This was a recent visit. And this was a man who came in concerned about a rash that had developed in his groin area. He's a man in his early 50s, and the reason I'm seeing him is he's on pre-exposure prophylaxis for HIV. He's here for renewal of that, for his chronic HIV testing, but he brings in this new concern. And he tells me that he lives in an open relationship with his male partner, also on PrEP, I will say, and he describes that he had a sexual encounter with another male in New York City, that he sort of thinks there's a connection here. But then as we talk more, he is in this open relationship. So there are rather frequent sexual encounters with other men other than his live-in partner. He says this problem's been going on for a few weeks, and he describes a rash in the pubic hair area. He says it's very itchy. He says it's mostly around his umbilicus, and he seems to note, he makes a comment that he notices these little blue spots in this area. So there's a rash, but with little blue spots. And he actually bought a magnifying glass, and he was looking in this area and describes to me the things that he was seeing.
Got a lot of guesses on this one.
Okay.
First one's from Alex. Given how my guess went last time, I figured I would try again to see if I can do it again. So I have to say that I like how parasitic diseases is laid out, made it really easy to go to the back of the book towards the section I needed. My guess would be that the man in the city has crab lice, Therius pubis, or the papillon d'amour. Crab lice can produce a blue spot. They are often transmitted through sexual contact. Crab lice can be treated by applying a pediculicide to the affected areas. Yet another word for me to look up. A quick Wikipedia search led me on to seeing that ivermectin was approved for treatment of pediculosis, and that gasoline was also used to treat head lice, Pediculus humanus capitis. Resistance to drugs was also noted. Per parasitic diseases, permethrin 1% is the recommended treatment for head lice, which the book also said that head and crab lice can be treated similarly. Clothing should be washed and dried by exposure to heat 7 degrees Celsius for 30 minutes. Also, I would suggest that recent partners get checked and treated to prevent spread. Is there any chance for louse-borne diseases? On another note, every time I have looked for TWiP webpage, I have always gotten this week in photography. I have learned my lesson and just bookmarked your page. Yes, TWiP is a longer-running photography web podcast, but you could just go to microbe.tv/TWiP. I think that's pretty easy to remember, no?
Hopefully. Or just type in the whole "this week in parasitism" as opposed to parasitology.
Yeah, that would work. This week in parasitism. Yeah, it goes right there. It's the first hit. And then there are many more. Speaking of hits, by the way, Google now has a podcast app on its Android devices. And I've put links, if anyone out there has an Android phone, I'm sure many of you do, you can now click the link on the website and it will open in Google Podcasts.
All right.
Can you get diseases from lice, Daniel?
Some lice, yes. And so, for instance, the body louse is classically something that carries relapsing fever. So, yes.
Got it. All right. Take the next one, please.
Chris writes, "Hello, professors. It is a sunny and beautiful day at 82F with a slight breeze here on Long Island. I believe that the patient from episode 153 is suffering from a case of genital lice. Based on the symptoms presented and the fact that he is sexually active with multiple partners means that he could have easily picked up this parasitic louse that is known to cause itching of the genitals. Additionally, because you could see it with just a magnifying glass, leads me to believe that this is pubic lice. Another possibility for what it could be is also scabies. as this parasite can also cause intense itching of the genital area, although these tend to present with visible lines and also infect the hands. If I win the book, please let someone get it, as I already have a copy that I have displayed proudly in my office. On an unrelated note, I recently went sampling in Alaska for parasites, and I've attached a picture of the parasite Shistocephalus Solidus, because I thought you might all enjoy seeing how large this parasite is in comparison to its host. The tapeworm species has a 3-host life cycle between a copepod, the 3-spine stickleback, and some sort of waterfowl. Shistocephalus Solidus can make up to 90% of its host body mass. Truly fascinating. Warm regards, Chris. And that's amazing. He's got a photo here where you've got these parasites that are, well, they're about 90% of the size of the fish.
It's amazing, isn't it?
Oh, that is.
I guess it takes a lot of nutrients from the host, right?
Yeah, it has to be that size. That's amazing.
Yeah, Dixon, if he were here, would probably wax poetic about this.
Yeah.
But he's not. Courtney writes, Dear Vincent, Dixon, and Daniel, greetings from Omaha, Nebraska, where it's approximately 79 and cloudy. Our weather the past few days has been relatively mild. I'm a master's of biology at the University of Nebraska, Omaha, studying bee populations at Glacier Creek Preserve, a prairie owned and managed by the university. My guess for the week is Thyrus pubis, or the crab louse. I admit I'm a bit too lazy right now to do a differential.
Okay.
Good thing your physician doesn't say that, right?
Yeah, a little tired, end of the day.
Here's an interesting article I found on Reddit regarding malaria tests without using blood. Mr. Jita had been tested 4 times for malaria, with the first 3 reflecting negative results. He then designed and made a device that clips on the finger and scans the blood for changes. So sends a link to a paper in BBC News. A Ugandan made this device, and he won a prize. It's a bloodless test that somehow it clips on your finger, and it can sense the changes in the red blood cells caused by occupation by parasites. What do you think about that, Daniel?
You know, this is the kind of technology we talk about every so often, which is fantastic, because you go to these places and, you know, I give one of the President Bushes, one of the George Bushes, a lot of credit for all he did for AMFAR, malaria in Africa. But I think a lot of what has happened is with better diagnostics, we realized that a lot of things were being chalked up to malaria that really were not malaria. And I would say the easiest way to half the incidence of malaria in a region is to go there with a diagnostic test, and you realize that, oh, half the things are not malaria. So we really need diagnostic tests in these areas where there's problems with having skilled technicians, having equipment. And so stuff like this is fantastic.
Yeah, it's cool. Matibabu is the name of it. Matibabu, which means treatment in Swahili. It's amazing. The red beam detects changing the color, shape, and concentration of red blood cells. Well, I hope it works. Sounds great.
Oh, that is great.
Nice.
Peter writes, Bonjour, Professors TWiP. You commented on how my current working in Montpellier, France, may prevent the TCD parasitology group from sending in answers. Although we did not reach the time-synced approach of Bill Gates that Daniel suggested, rhymes with Twipper, it's Gwen Dislipper, and I did email back and forth on what it might be. Vincent is right, though, that it is a lot of fun trying to solve the case as a group. It has also been a good excuse for us to all get together, as we are in different offices throughout the building. I will be traveling for a few weeks, but when I get back to France, I will see if I could get people here listening with me before I return to Ireland. I'm not sure if an Irish guy, they don't know very well yet, saying he has something interesting for them to listen to, and then playing 2 guys talking about pubic lice would be the best first impression, though. So, yes, I believe the gentleman has pubic lice, or the crab louse, due to the characteristic blue spots. Blue spots resulted in the ruling out of strongyloides, but I am curious if Daniel knows of any other pathogens that might cause blue spots. Lice can be confirmed by trying to comb them out and viewing under a microscope. According to PD's 6th edition, treatment with dusts, creams, lotions, and shampoos. Also, heat treating any sheets, combs, clothes, etc. that may have been contaminated. Also, I know at TWiP you are very interested in science communication. I thought I should mention labmate Maureen Williams, who did a great job winning many rounds of the FameLab science communication competition, championing parasitology all the way. Here's her national final talk on protecting parasites. Bientôt, Peter Stewart at TCD Parasitology.
Nice.
All right.
Any other parasites cause blue spots or pathogens?
Well, I'm worried that if we say that, it might give away the answer.
Yeah, we'll wait. We'll wait on that. Sure, no problem.
Okay.
Next one is from Eric. Hello, TWiP hosts. This patient's history of recent new sexual contact, intense itchiness, and seeing things like moving living lice all suggest that he has acquired pubic lice, Thyrus pubis. But as a habit, I should also consider what else it could be. A very common cause of itch in the groin would be tinea cruris. Tinea is the growth of any of several fungus species on the surface of moist skin, including athlete's foot and ringworm. When tinea occurs in the folds of skin at the groin, it is called tinea cruris. However, I posit that the presence of the itch, even in abdominal hair, hints that the rash follows the hair, not the moisture. Also, blue spots are not associated with tinea, I read, but describe pubic lice bites. Lastly, I'm sure there are non-infectious itchy rashes in the groin as well, though I do not know any off the top of my head. Should our patient be screened for other sexually transmitted infections? And a question that popped into my head, do we know how common pubic lice is nowadays compared to 50 or hundreds of years ago? Do the lice taxa that evolved with our species now qualify as endangered insect species, or are lice far from eradication even in developed countries? Thanks again for the intriguing cases, Eric, Los Angeles. Good questions, right?
Those are excellent. I'm going to make sure that we go through and answer all these. I wonder if everyone's going to have the same answer. Daniel writes, yo peeps, my guess is that your patient with itchy private parts has pubic lice, the organism. And how we pronounce it? Where's Dixon to help us how to pronounce it?
Well, I think the P is silent, right? So it would be Thyrus.
Thyrus pubis. I don't know what people do when they learn our language. Like, the P is silent. So anyway, the organism Thyrus pubis is a blood-feeding obligate ectoparasite of humans. The lice are transmitted by close physical contact and occasionally via fomites. They live on coarse body hair. As described, the moving crab-like insects are visible with the aid of a simple magnifier. The blue spots, or macula cerulae, are caused by intradermal hemorrhage from louse bites. The itching is caused by delayed type hypersensitivity to the saliva and may take 2 to 6 weeks to develop. Treatment is with topical permethrin or malathion. This does not kill the eggs, so treatment is repeated after 10 days to kill newly hatched survivors. Bedding and clothing should be washed at high temperature. His partner and other recent sexual contact should also be treated, although Daniel alluded to the difficulties with contact tracing when people use an anonymized app to hook up. I was interested to hear about Daniel's forthcoming visit to the Global Health Program in Glasgow. I go up there a few times a year for an exam committee at the Royal College of Physicians and Surgeons. I don't think I'll be in Glasgow in January, but if he becomes a regular visitor, our paths might cross in IRL. I also have the hardback 6th edition, greatly received, so please leave me out of the prize draw. As ever, thanks for an unmissable podcast. Daniel, London, UK.
I love it. An unmissable podcast. Wonderful.
So I should comment. So I said global health, but it's the University of Glasgow Diploma in Tropical Medicine and Hygiene course that I will be teaching at.
Is it the same college, Royal College?
It's the University of Glasgow.
University, got it. Isn't it cool that there's another college of physicians and surgeons?
I wonder if there's some connection in the naming. I would suspect, right? When they were naming Columbia, the Columbia College of Physicians and Surgeons, they
maybe.
I think they're trying to follow in a British tradition.
Yeah, it could be. It could very well be. All right, the next one is from Trudy, who writes, Dear TWiPers, the guy from TWiP 153 has a crab louse infestation caused by Thyrus Pubis crab lice are most frequently transmitted by sexual contact. They are known for causing intense itching and producing the characteristic blue spots identified by the patient. Infestation can be treated with both over-the-counter and prescription medications. Common over-the-counter medications include a louse-killing lotion containing 1% permethrin or a mousse containing pyrethrins and piperonyl butoxide. Piperonyl. A second round of treatment is recommended within the following 7 to 10 days to kill newly hatched nymphs to prevent reinfestation. It is usually recommended to wash clothing and bedding, followed by drying at high heat. Shaving pubic hair may help as well. Lastly, I would particularly recommend staying away from the person who caused the infestation to begin with. Monogamy may reduce risk too. Just a thought. It certainly might, right?
Kindly regards, Judy. I like that. All right. Octavio writes, Dear L3.
Oh, that's an interesting appellation.
I like that. The symptoms and signals manifested by the man described in the last case should be caused by an infestation of crab louse, or in Portugal, we call these chatos, and he pronounces these, chá, as in like Hugo Chavez, and tos, as in risottos. So it'd be, I guess, chatos. Chatos can be translated as someone or something immensely annoying. Pubic pediculosis is usually sexually transmitted, but can occur after contact with fomites, such as clothing, bedding, and towels used by an infected person. Treatment can be performed by an oral dose of ivermectin, topical application of lindane or permethrin. In Portugal, topical products with permethrin are no longer available for human use. Therefore, the affected have to choose one of several over-the-counter products for head and body lice containing dimethicone, a kind of glue that covers and desiccates the parasite. In my work as a trainer for pets, ectoparasitic products, I've learned that in some places, fipronil sprays for treatment of fleas or ticks in dogs and cats are very popular for the resolution of this kind of problem. Users only complain a little because the spray's alcoholic solution burns a little bit, probably because the skin is more or less severely scratched due to the intense itching. Thank you very much for your invaluable service teaching, informing, and spreading the words of science. Please, please carry on. Octavio from Portugal.
It's nice. Thank you, Octavio. Appreciate it. Alice writes, Hello, TWiP team. Imagine my delight upon returning from Canada to find my signed copy of PD6 on my desk. Thank you very much for this beautiful book, which I will reference here for my guests for TWiP 153's case study. I'm originally from Maryland, and we have a notorious T-shirt that says, We have crabs. Well, perhaps this gentleman should procure one of these T-shirts for himself, since I'm pretty sure he does in fact have crabs. Theros pubis, to be exact, obtained through sexual contact while in the city. According to pages 472 of PD6, crab lice produce characteristic blue spots, which are noted on this patient. Besides the intense itching and visualization of the papillon d'amour with a magnifying glass, the patient is otherwise healthy. Treatment is topical insecticide in the form of a lotion, cream, or shampoo. For crab lice specifically, PD6 mentions applying pediculicide and repeating after 10 days, as well as frequent clothing washes and changes. Dry cleaning bedding is also recommended. Cloudy and rainy here in Syracuse today with a cool 18.9 cm. Looking forward to more TWiP episodes, especially now that I have such a great reference to use for the case studies. Alice from Syracuse, New York.
All right. Kevin writes, Chicago weather, wettest May on record, or near enough. Illinois reports its first West Nile case. The subacute course of few weeks and largely external nature of the symptoms should restrict our speculations to skin conditions, dispensing with lengthy differential diagnoses that would include skin manifestations of systemic illness. The patient has not had any woodland exposures, having confirmed his activities to cosmopolitan locations. The presence of blue spots, also known as sky blue spots, or macula cerulae, a further note on this below, inexorably drags us into the lair of the crab louse. Thyrus pubis. I'm waiting for Dixon to correct me on this, right? Where is he when you need to be corrected? Also formerly known as Thyrus inguinalis. The generic name is derived from the Greek Thyrius, to be infested with lice. I think that would be Theriasis.
Yeah.
A quaint description of this pest is seen in John Stevenson's 1831 Medical Zoology. This disgusting parasite inhabits the eyebrows, pubes, and etc. of men and women. It is a frequent cause of local prurigo. A less direct and more Victorian description was found in 1867 English Encyclopedia, where the pubes are only suggested by a prim, etc. And lastly, the pubis of Linnaeus, which constitutes Lake's genus Thyrus. This species inhabits the eyebrows, etc. is commonly known by the name crab louse. The crab louse is about 1 millimeter long, very dorso-ventrally flattened, and rather translucent in appearance, making initial detection a bit difficult. The obligate bloodsucker keeps a tight grip on the hairs and lives closely adherent to the skin, which can mimic a small scab or excoriation. The pubic hair, male escutcheon, eyebrows, and eyelashes are common habitats, and many other hairy areas can be incidentally infected. I've seen them on the ventral aspect of the forearm. A reference is appended that describes a scalp infestation. Transmission is almost universally person to person via intimate contact, but some wiggle room is afforded for afflicted partners in monogamous relationships by rare reports of transmission by fomites. I was amused to see that PD6 still invokes the legendary toilet seat defense as a mode of transmission. I will suggest that the conventional smooth plastic toilet seat is a very unlikely launching pad for the clumsy wingless louse. However, in defense of PD6, a lovely book which I consult frequently, I have appended a photograph of a plush fabric toilet seat that might answer to the needs of a wandering crab. I should, Dixon and I had a little discussion about this. I am of the belief that toilet seats do not transmit pubic lice, but for some reason Dixon got a little emotional when I suggested that we. He seems convinced, and we'll have to ask him when he comes back why he's so convinced. But anyway, I think I would say, in my professional opinion, we don't really think that the typical toilet seat is a wonderful place for these organisms to exist and transmit from person to person. All right. Crabs reproduce. So I agree with our emailer. Crabs reproduce on the host and lay their eggs on the hair shaft. According to the Natal, 1917, the nits of pedunculus, body, and head lice differ from Thyrus nits, but this is only of academic interest. Unlike the human body louse, Pediculus humanus, which can transmit Rickettsia prowazekii, this is epidemic typhus, or Bartonella quintana, this is trench fever, and Borrelia recurrentis, relapsing fever, the crab louse transmits no known illness. I agree with this. In the early days of the HIV epidemic, there was a great deal of interest in the theoretical transmission of HIV from human ectoparasites, lice, and bedbugs, and other blood-sucking insects. Some of the early speculations and research on this sphere may be seen in the 1987 Office of Technology Assessment monograph, Do Insects Transmit AIDS? Treatment in the early 1800s might include mercuric oxides. Fortunately, now one can go to the local pharmacy and pick up the 70s favorite A200, piperonyl butoxide, and pyrethrins, or RID. These agents are not ovicidal and so require retreatment in 7 to 10 days. Scrupulous adherence to package directions is essential for complete cure. Eyelash infestation managed by manual application of the insects or the use of ophthalmic-grade petroleum ointment applied 2 to 4 times a day for 10 days. Everyone seems to love ivermectin, but 4-ounce tube of the topical form costs $280. I would like to close with a note on the curious blue spots, a phenomenon that I first heard of on TWiP 153. A fascinating article in the journal Parasitology, March 1924, Maculae, cerulae, and Thyrus pubis. The microdissections of crabs and bizarre human experimentation, presumably done on Russian military recruits, are a glimpse into medicine's sordid past. A quote from the article: "The blue spots induced by the bites of Theris pubis in man's skin are due to the action of a ferment emanating from the bean-shaped salivary glands of the insects. This ferment acts apparently upon the hemoglobin, which, becoming discolored, causes diffuse bluish coloration of the skin to appear in situations where the insect injects its saliva. Thanking you for all your sublime educational service." And then we have a list of the references and, most importantly, a beautiful blue plush toilet seat, which must have been what Dixon was thinking about.
Yeah, maybe he has one, right?
That might be. I've been to his home and I do not remember a plush toilet seat
I
like
don't
that.
Either.
I'm not sure how hygienic that would be, right?
No, it's not a good idea. It's gonna capture. I mean, let's face it. When men urinate, they stand, and the seat often gets, especially with kids, it gets on the seat, right? And you can wipe it off. But if you have a plush seat, if this is too much for anyone, I apologize. But we are, Daniel's a physician; he's used to this, and I'm a scientist, and I'm used to gross things. But,
No, it's a bad idea to basically put a little bit of like plush rug on your toilet seat. And you're going to sit there. It's going to get contaminated, and someone else is going to sit there, and it's going to stay moist, right? Because of this issue with men having poor aim.
Yep.
It's, unless it's a single-use plush cover, which, that wouldn't really make much sense either. We recommend against.
I would, and it also encourages you to sit there too long, which is not a good idea because it's comfortable.
That's funny.
The sign. Have you seen the sign in the restaurant bathrooms? We aim to please, you aim to please.
Mm-hmm. Yes.
I just want to point out this word subacute in the first paragraph, which of course means something between acute and chronic, right?
Yes.
It's a very interesting word. And in case people don't know, that's what it means. Because we aim to educate here on TWiP.
Yes.
The next one is from Lauren. There's just two more. Lauren writes, "Hello, TWiP. Case study for 153, gentleman from the clinic. My diagnosis is Theris pubis, pubic lice or crabs, and blue spots, a quote from Parasitic Diseases. Diagnosis depends on identification of lice or eggs. Eggs must be identified by microscopy, but it sounds like you got a positive ID of the lice. Treatment may be hard and tedious to the risk of reinfestation. When speaking with the gentleman, stress the importance of informing and treating the domestic partner as well. Simply washing affected areas with soap does not kill lice or nits. All current sexual partners and subsequent partners of both gentleman from clinic and his domestic partner must follow regimen of treatment or be excluded from sexual encounters due to the risk of reinfestation during infection. It may be easier to treat if closed relationship during infection. Treatment: over-the-counter treatment, prescription shampoos, lotions, creams with low concentrations of insecticides, permethrin, benzene, hexachloride. All bedding, towel, clothing must be washed and dried on a hot cycle daily, and clothing should be exchanged for clean clothing after every application. All daily grooming equipment heat treated, 70 or 158 for 30 minutes. Knit combs with closely spaced teeth to scrape nits. Eyebrows and beard need to be examined for pubic lice. If found, a thick layer of petroleum jelly will bring the lice to the surface and then must be pulled out with tweezers. Do not use insecticide on the face. I recommend for our gentlemen knit combing, oral ivermectin, and or benzene hexachloride for second-line therapy due to persistence of lice, and concerns regarding benzene hexachloride toxicity for his domestic partner. Permethrin 1% is recommended. Knit combing. Gentleman and his partner could treat each other, allowing for more accurate treatment. It is important to be thorough because most treatments do not affect eggs. Pubic lice can produce 30 to 50 eggs. The egg stage lasting around 10 days, lifespan 3 to 4 weeks. Adults need a daily blood meal. Nits do not until they are hatched. Goal is to disrupt the life cycle of the lice before they can lay eggs. Bedside manner is also important so he does not feel embarrassed and hopefully will come in for treatment for STIs earlier next time. I know you guys are truly caring doctors. Thank you for taking time for making TWiP. Well, Daniel is the truly caring doctor. I am the scientist. I care about certain things. But I don't know if it would call me true. I truly care about viruses. Yeah.
Yes. So I'll read this last one, then we can run through and respond. Cullen. So here's our friend. Are we pronouncing it Cuellen now? No, Cullen. Cullen writes, "Dear TWiPers, my guess for the man with itching pubis and umbilicus is Thyrus pubis, commonly named crab louse. Given the distinct wounds and constant itching, I would say these bugs are living at the base of his pubic hairs and consuming their blood meal, causing small wounds that can be bluish in color. As always, thank you for a fun podcast I can listen to while pipetting and plating. Sincerely, Cullen in Atlanta, Georgia." Okay, that's it, right?
That's it. That's all the emails we have.
Well, we seem to have a consensus here. Unless, Vincent, did you want to throw out some different diagnosis?
This is the one that popped to my head the first time I heard it, for sure. But I like that the one person thought of some fungal infections, right?
Actually, and I think that was actually good that someone brought that up, because there was someone who asked, are there any other things that make you blue?
Yeah.
And actually, there are certain fungal infections that present with this bluish color. And so it's important, if you didn't have the right clinical story, to start thinking about those things. Or let's say you treat it and you say, hey, this isn't getting better. So there are certain fungi that can present like that.
Yeah. I also like that, early on someone asked whether lice could transmit diseases. And then I think it was, gave us a nice list of, I think that must have been Kevin, right? He said, yes, the human body louse can transmit rickettsia, trench fever, and Borrelia, but the crab louse transmits no known illness.
Yeah.
So thank you for that.
No, and I think that's an important thing. So I will go ahead, as we go through. First, I will go ahead and say yes, the consensus got it right. Mm-hmm. And how do we know? So one of the first things is this was a pretty compelling story. I mean, when this person comes in and tells me the story, I mean, we have the appropriate clinical context, as we like to say. So this is a gentleman who is not monogamous, who is having close sexual intimate contact with multiple partners, particularly in New York City, which is an area where we see this. He describes this itching, which is fairly classic. And then the blue spots are quite something. And I thought it was great the way, I think it was the reference to the Russian research, which I wonder if we're even supposed to really mention when stuff is done inappropriate like that. But it actually, when the lice are feeding, they actually inject, and it's a component of their saliva that causes these little blue spots. So the people will often describe these little blue spots, intense itching, and they may even notice like, hey, I see stuff in my underwear. And these can actually be the feces. They can also be bleeding that gets sometimes if it's a really bad infestation, they'll actually have pretty significant swelling of the lymph nodes in that area. So let me see what the other things I thought. Oh, we should talk about bedside manner. Appreciate that. And I think that's one of the things, sometimes, particularly maybe for me, I can enjoy the diagnosis, more than I probably should. Shall I say that? So I know when this person was telling me this story, I said to them, you see me every 3 months for your PrEP renewal, but if you have a problem, you can come and see me more often. You don't have to wait for 3 months. So I always try to, it's important for them to feel like they can come and talk to you about these things. So that is important, because there can often be I think, opprobrium. You can send this message that like, oh, well, you got this because you did something, and this is a consequence of that bad decision. But, I try to be non-judgmental as a physician. And so I try to just let the patient know this is the exposure. This is what put you at risk for this. And then try to make them feel like this is sort of a safe space. They can come and tell me these things. And I think then hopefully they're telling me more things.
Yeah, for sure. It's only if it's for their own good, right?
Exactly. Well, it's for everyone's good, right? I mean, let's say this gentleman didn't tell me about this, right? He was embarrassed or whatever, or I had horrible bedside manner, which maybe I do. Who knows? I try not to. He would then continue to transmit this, right? As mentioned, he's going into the city periodically in this open relationship. His partner is also infected too, probably. And also, so, you've got this whole infectious epicenter that, hopefully you're going to intervene, and a lot of people will benefit from his just being comfortable sharing this story.
Sure. Of course.
The magnifying glass. So that's, I think, important here. I liked that he actually went and bought a magnifying glass. The size of these little critters, they're only about 1 millimeter. So you're really not going to see these with the naked eye. So you do need some ability to magnify. Right. And so you can either do that with magnifying glass, or a lot of people have in their pockets these smartphones. Mm-hmm. And a smartphone camera can often get a nice magnification. And so you can take photos and magnify and actually see these without having to go through a lot of expensive diagnostic testing. And so that's something I actually do quite a bit in the office and in the hospital. Is I'll use my phone to take photos, then magnify to get a much better view of what's going on.
Right.
Let's see what else we have. Okay, so I'm glad that this is not a parasite that occupies or becomes 90% of its host's size. So we can pass that.
Truly.
Thinking how frightening that would be. Now, it's interesting that someone brought up strongyloides. Yeah, I was not at the top of my differential here, but they do, when they normally talk about the differential here, they'll throw scabies in there. I think some of our emailers did, and that's a good one. And those will not tend to have the classic blue spots. They'll have these streaks, but they'll have the similar itching.
Right.
And they'll have these linear brown lines, and particularly at thermoscopy, if you get in, take a look, you can see the characteristic changes there. Some of the I think coronary bacteria and some of the other fungal ones can be issues here. Mm-hmm. But I think in this case, you put together the exposure, the blue spots, and we're actually able to visualize the crab louse, with magnification. So that was, I think, helpful. Asking about other STDs. Now, this is a gentleman that sees me every 3 months for his prep. So we routinely screen him for HIV, for gonorrhea, chlamydia, syphilis, hepatitis B and C. But should this be a patient you see outside of that sort of routine, I think it is really important to realize that this is a sexually transmitted infection in general, with deference to Dixon and his toilet seat. And so you want to look for other things. But that is interesting. Someone brought that up too, is that in let's say a wet, stringy environment, like a towel or there are certain fomites, and people can get this without it being a sexually transmitted disease. So I will say that it is not always a sexually transmitted infection, but it is often and usually. Mm-hmm. The fact that it takes a while before the itchiness develops, that was another thing too, because this man had sort of pegged a sexual exposure and said, oh, this is when I caught it. Mm-hmm. But the intense itching, the hypersensitivity that drives that, takes several weeks to develop. So this may not have been the sexual exposure that he attaches significance to. It may have been a sexual exposure a month and a half before.
All right. Interesting.
Shaving the pubic hair is a good idea. Interesting. I think a lot of young people do this as just part of a social thing. Mm-hmm. There's a lot of shaving of pubic hair, but the lice grab onto the hair shaft, and the diameter of the hair shaft is important for a lot of the different lice species. Yeah. And but you have to really, in this case, cut the hair very short. It's really a shaving because they're right up against the skin, and their little nits are also right up against the skin, so they're much harder to identify, and you really have to complete hair removal to get down to where the nits are.
And that would take care of the infestation without any drug treatment?
If you were in a situation where you could completely shave, it could be a treatment option. If you completely got rid of the hair, you would basically remove the habitat. But I think, as people mentioned, it can not only be in the pubic hair, but these little guys can actually get into the eyelash the eyebrow hair. So sometimes you see them outside the pubic hair, in other areas.
Okay.
So I don't know, for some reason I find that very unpleasant to think about. So
yeah, eyelashes right above your eye there, right?
Yeah,
that's weird.
So, not exactly. So eyebrows mainly, but eyelash is also a possibility. So let's see what else we have to say. Oh yeah, the fact that we don't have to really worry about it's just this unpleasantness. We don't have to worry about other things. What else? Treatment options. So a lot of it's nice that people mention some of the non-pharmacological approaches. A lot of times, particularly for eyebrows, people will use a thick Vaseline because you don't really want to use some of these toxic things. You really don't want to be using a lot of these toxic permethrins, pyrethrins, etc. on the face. We don't use Lindane really much at all because of concerns of toxicity. And when you're having a trouble getting rid of it, sometimes we will also resort to our good old ivermectin. So, and then of course you've got the whole ring control issue. It's not just this man, it's his partner, it's his other sexual contacts, and it's probably on a lot of stuff in the house. So he's got to let his partner know, hey, partner, we have an issue here. We've got to wash all the clothing, all the bedding in warm, hot water, we say greater than 70 degrees C. And you've got to treat initially, and then often your treatment won't actually destroy the eggs. So think of this almost like a pinworm thing where you're treating, and then you're treating again in about two weeks, 10 to 14 days, we say.
Do you think, speaking of PrEP, do you think that giving people PrEP gives them a full sense of security? They don't think about other sexually transmitted diseases?
I don't actually, and I don't think the data would suggest that. A lot of interesting things that people have. I remember there was an idea recently that someone was discussing with me with HPV vaccination, and they said, "Oh, now that there's HPV vaccination, all these young women will have much more sex."
Yeah.
Not true. And the people that I tend to see for PrEP, this man is not a poster child for this comment, but in general, they tend to be concerned about sexually transmitted diseases. And so
I read a study about the HPV situation where they had taken, I think, a number of young women who were sexually active and followed them before and after HPV vaccine, and it didn't change their behavior. Most of them were practicing safe sex and so forth after being vaccinated.
Yeah. And
these were sexually active women to begin with, and I wrote a blog about this, and someone commented, "Well, that's the wrong population. They're already promiscuous, right?" But I'm not sure that's a valid argument. I mean, maybe people who are looking for an excuse not to have their kids immunized for HPV would use that as an argument that, that's an invalid study because you got that population. But I think you're right. And you would know, you see these populations, and it's more of a, I don't know why parents would think that getting an HPV vaccine would make them more promiscuous. I mean, I heard that from my kids in school, from other kids all the time.
Yeah, it's an interesting idea that, I have 3 teenagers, and to think that what keeps them from being sexually active is a fear of some virus most people have not heard of. And then once they get this vaccine, that fear or whatever is holding them back will be unleashed. But it seems, if anything, when and you look at these studies, so the studies definitely don't demonstrate that there is not an increase, and if anything, there's an educational aspect. Letting them, you know what, your parents may be focused on pregnancy issues, but there's a lot of risks to becoming sexually active if you're not careful. I would say there's no such thing as safe sex. It's just safer sex. But there's HIV. We have over 1 million people in this country who are HIV positive. So the PrEP is an excellent idea for just about anyone who's sexually active and not in a monogamous relationship. Even people who think they are. Unfortunately, we had a recent woman, 3rd trimester, got infected from her husband, who she thought she was in a monogamous relationship, but apparently the husband was not. So it is tough. But I think it's important for people to realize all these risks are out there. And, this is probably getting pubic lice is probably the least of the risks compared to all the other risks out there.
All right. What did you end up using for treatment in this case?
So in this gentleman, we used combination therapy because he'd been using a lot of things over the counter that hadn't resolved it. So we did the permethrin, the 1% permethrin cream, and we also gave him ivermectin, and we did a dose, and then we did a repeat 2 weeks later, and then did the whole regimen. And he did well. Finally, he was quite relieved when I saw him next.
All right, very good.
I guess two more things I should throw in.
Yep.
So someone mentioned West Nile virus. Where was it where they had their first
Illinois,
case?
I think. Illinois.
Back when I was living in Colorado, I actually diagnosed the first case of West Nile out in the state of Colorado. It's very exciting for me.
Sure.
For me. It was back when it was hard to diagnose because it was just coming into the country. So you had to work with the CDC. There was no commercially available assays.
Now I should say that it must be from this season because Illinois had seen West Nile way back when. Okay, so that's got to be this season because I'm looking at a page from the Illinois Department of Public Health. It said West Nile was found in 2 dead crows in the Chicago area in 2001.
Yes, it must be the first of the summer, first of the season. But I should also, so now that we close out this case, I should give people I got some follow-up, right, on the cases from Panama. And I think, let's see, we had the woman who had the abdominal pain that we thought was giardia. We treated her. She's the woman with the ovarian cyst.
Right.
And a couple things happened. One is she actually got better, which is great. The other thing is in the interim, right, every 3 months they're going there, she had the ovarian cyst ruptured. But she went on to do fine, repeat ultrasound. It was all good. So she is much better. She was that poor woman with a really long period of abdominal pain and the 2 itchy kids, and the 2 itchy kids are also better. We also had the gentleman who had the bloody diarrhea, and he also resolved with treatment, which we targeted for entamoeba, right? So amoebic dysentery.
Right.
So it was nice to hear everybody did well.
Very good. Excellent.
Yeah.
All right. We had 12 guesses. So let's see. Let's give away a book. Now, it seemed like a number of these individuals already have the book.
Oh, no.
I wonder if we're going to get a point where everyone has a copy already and it's the same people, over and over again. Well,
what we could do is if you've already won and you write in and you win a second time, if you want, maybe you can, we should probably put this on the website, but we have a list, right, of all the places we've donated books. Maybe they can find someone in another part of the world that we have yet to donate a book to, and in their name, we could make a donation.
Yeah. All right, 13. Is it 13? No, it was 12. Let me get a random number between 1 and 12. Push the button, number 7. So let's see if number 7 had already got one. Number 7 is Trudy. Trudy. Now, hi, Trudy. I know Trudy. She listens to TWiP. I saw her in Atlanta. Don't remember if you have parasitic diseases or not, the signed edition. So, we'll have to record this and let us know, Trudy. And if you didn't win it, send us your address to TWiP@microbe.tv. And if you did, just let us know and we'll give it to someone else. Now, last time we redid the drawing because the winner had already had it, and Sue Ellen won, but we haven't heard from Sue Ellen. So I need your address if you want to get the book.
Certainly. And
Cullen, I owe you a book. I haven't mailed it yet. I've been traveling a lot, and I will not mail it until next week when I'm back, because tomorrow we're off for TWiP 500 in Texas. Now, today what we thought we would do is email, because we have not read an email other than case guesses in many, many weeks. And now we have quite a few here. We'll do as many as we can. And some of these are actually old case guesses that were behind. They came in so late that it just doesn't make any sense to read them as part of the case guesses.
Okay.
First one is from Lawrence, who writes, Dear TWiP team, I'm a regular listener, but unfortunately I'm typically several weeks behind on listening to my podcast, so unable to venture a guess case study. I had a more general question that I hope you can help with. How do you stay current with the literature? I subscribe to newsletters from a handful of journals and organizations, but I imagine that there's a lot of research that is not covered in these sources. Is there something like an infectious disease digest that summarizes recent research? I've tried searching with Google for something similar, but no luck. How do you stay on top of current research? I enjoy listening to the TWiP podcast, but hope there might be a regular publication that highlights notable research and advances in the field, which might also include things outside peer-reviewed publications. Well, Lawrence, what I do is to subscribe to all the RSS feeds from each journal. Every journal, now an RSS feed is simply a way for finding out when a web page changes, right? You probably know what it is. If you go to a journal website, they will have somewhere on the page a link to an RSS feed. And then you use an RSS reader. There are tons of free ones out there. I use one called Feedly. I know Google used to have one, but they stopped doing it. And I put the RSS feed for each journal into this, and then it gives you a nice list of all the new articles by journal. And I simply go through those, and you can do it on your phone. And if I'm waiting in line somewhere, I'll look through. And then you could star articles that you want to come back to later. And I often do that for the podcast, and I pick articles that way for doing that. I find it's the best way to keep up with many, many journals, because there's no one source that is going to summarize everything. Obviously, it's just too much out there. It's always going to be a selection, and you might miss something. So this way, I first have to decide what journals I'm going to look at. And there's not that many. The ones I want to look at, maybe 50 to 75, virology, microbiology, infectious diseases. And I find it works. You have some way of doing it, Daniel?
I agree with the people who feel like the amount of information is now somewhat overwhelming. So I actually have a number of journals that what I'll do is I'll skim the basic table of contents.
Yeah, sure. And
so the three IDSA publications, the Parasitology Society journal, some of the tropical neglected diseases. So I actually probably have about 20 that I main, and then I have a few sort of search terms on PubMed where, boom, when things get published, I get a little more. But it is a challenge. For those who podcast, I actually like the Puscast by Mark Crislip. Mm-hmm. Which I think he does a good job for clinicians. Of every couple of weeks he comes out with, he's reviewed a lot of literature and he gives you little tidbits. And if you find something you say, I want to read more, you go to his webpage, EdgyDoc, and you can then take a look at the articles. Some of them are behind paywalls, which I think is unfortunate. But no, it's a challenge to stay up to date.
Yeah, Mark's Puscast is great. Of course, it's his selection, of course, and
that is true.
And sometimes I find an article there. In fact, the HPV vaccination sexual promiscuity result that I just described, I heard him first on Puscast, so I'll pick up things that I don't normally look at. But if you want a really unbiased selection, I would say RSS feed. I used to have all the tables of contents emailed to me, but that got ridiculous. I never looked at them because there were too many, right?
Yeah.
But an RSS feed can sort it out in a better way. Anyway.
It's a challenge. I like the RSS feed approach.
All right. Why don't you take Anthony's, please?
Anthony writes, cannibalism was far from unknown in our species' history. Through this route, polio in the CNS would not be a dead end. Polio disabling a host that then would be easy prey would encourage transmission. Balis ascaris larva migrans cripple small birds and mammals and so makes it easier for raccoons to catch them. My polio have so helped its favorite hominids to have their neighbors for dinner. Okay.
So this refers to a discussion I made of the CNS being a dead end for viruses because they can't get out. And yes, and this is for humans only, because a virus in an animal that got into the CNS could still spread to another animal because animals eat each other, but not humans. And yes, cannibalism, right? So there are two issues with that. It's a perfectly reasonable idea. First, we're not sure when polio infected people, and might have been later than when humans were involved in cannibalism, right?
Mm-hmm.
Although I know the 4 people of New Guinea were involved in cannibalism until not too long ago, right? But that's just one.
There may still be a little going on.
Yeah, but it wouldn't explain global.
True.
Persistence of this virus going into the CNS. And the other is that, let's say that polio transmission was selected from the CNS by human cannibalism. So that could be, and that still persists because it's been too short a time to get rid of that. So this is a supporting idea for that theory, because we have lots of features. For example, when we were hunter-gatherers, we evolved certain features like the ability to go starving for weeks at a time, right? And having low-salt diets and so forth. And this is why now we have an obesity and diabetes epidemic, because we still have those genes that enabled that kind of hunter-gatherer lifestyle. They haven't gone away. I don't know if you have read Dean Goldman's book, Daniel, where he talks about all these traits that we had years ago that are killing us today, essentially, including obesity and diabetes, because they're based on features that are no longer an issue. We don't have to hunt for our food. We don't have to starve for weeks at a time. We have too much salt in our diet and we're not used to it, and so forth.
Yeah. Is this the Too Much of a Good Thing? Is that the book that?
Yeah, I think so. Yeah, it's on my list to, I think I downloaded a sample to read it. But for polio, again, yes, it may have been selected for by cannibalism. And that it hasn't been long enough for that to have left, for the virus to have evolved to lose it. So that's that. The next one's from Noah, and he sends a picture of page 533 from Parasitic Diseases. This page is inexplicably confused about the singular nucleus and plural nuclei. There must have been some sleepy copy pasting. So that's for you, Daniel.
It's all Dixon's fault, and I have actually gone through and fixed all these. So we receive funds. We're going to do a third printing, but we're also going to get professional editing, and we'll be coming out with new PDFs and new Kindle versions, and all of this will be fixed.
Right.
But yes. So thank you. And actually, you go through and you find stuff in our book, email us. I am like that man with the scratchy groin, just waiting for you to help us.
All right.
So, hello again, TWiPitos. This must have been my month to find these articles. Have you seen this one? And let's see what we've got here. This is a couple infected with parasitic hookworms at beach sharing horrifying photos. I remember this one, actually. I have to say, they're good pictures. If people follow these, because you normally have this idea that you're going to see under the skin, much like some of the pictures in our book, you're going to see the outline of some serpiginous parasite, but you can often end up with these blistered areas, which is an immune response to that. And so if you look at some of these pictures and below them, you see a bottom of a foot, it looks like this blister area. You think, what could this possibly be? It looks like, a horrible infection between the toes. And so these unfortunate individuals were probably on a beach. They stepped somewhere or stood somewhere where there had been the feces, usually from a dog, sometimes a cat. And then there's aberrant entry of the parasite from the bird or the dog, and it's from the dog or the cat, and it's lacking the collagenase that allows it to go in deeper. So it ends up wandering on the surface until finally it dies. So what can I say? We grow them. That is me. Weird up here in the Northwest where it is 43F at 1 a.m. and no rain, but it is cloudy, of course. So we in Seattle miss the super blue moon. Okay.
Courtney writes, Dear Professors, greetings from Omaha, Nebraska. It's 32 Fahrenheit and nice enough to wear just a sweater. I hail from University of Nebraska, Omaha, where I'm a master's student. My research involves bees, and if they prefer a homogeneously managed or patchily managed landscape. I've been a listener for over a year, but this is my first time emailing. One of your recent podcasts, 146 or 145, you spoke about polyploidy and its prevalence. I thought I would point out that plants can get all sorts of freaky with their genetics, and many times have much more than 2 or 3 chromosomes. For example, Celosia argentea and Spartina angelica have 12. Fun fact, platypi, or is it platypuses, have 10 sex chromosomes.
It's platypuses. And I'm going to say that because it's Greek. It's not Latin. So you don't want to use Latin pluralization conventions when you're pluralizing a Greek word.
And then
okay.
This is, platypuses. On to my first ever guest. My method of narrowing down possibilities was opening your lovely book and control effing ulcer. First thing to pop up was cutaneous leishmaniasis. And I guess this was from a couple of weeks ago. I don't remember. None of which describes our patient. Acanthamoeba was a possibility. The last remaining were dracunculus, but our patient is not one of the countries. Tularemia was out. So I don't know. I guess it was cutaneous leishmaniasis. This sounds like a recent one.
I think that will.
I would really like it if you did more arthropod and fungal parasites. It would also be nice sometime to hear more papers about those things and not just from a molecular side. Thanks for the wonderful podcast. Yeah, we try and mix it up.
It's an interesting issue, like do fungal parasites, right? Because they are eukaryotic. So should we throw a few fungal cases in periodically? That, I would be comfortable with that.
That's
it
fine.
Might be a little outside of Dixon's wheelhouse. I'm not sure how you feel about it, Vincent.
I'm okay. I think it would be great. I mean, this is parasitism. It doesn't have to be worms and amoeba, right? And protozoa, I should say. It's fine with me. And
all
so
right.
now we're 2-1 against him. So
yeah, and he's not here.
He's not here, so we'll do it.
Be warned.
No problem.
I have some great fungal cases. All right. Luke writes, Dear all, I'm a first-time listener, recently introduced by one of my friends studying biology at Aberystwyth University in Wales. I first thought of guinea worm, dracunculiasis, primarily due to the presentation on the foot and the waterborne nature of GWD, as the rafting seemed key. After checking symptoms, distribution images, it doesn't seem to match up. GWD is restricted to Africa and India, where the patient has only been in South America. GWD is also noted as having a range of symptoms such as fever and inflammation on infection and allergic responses later due to responses to worm secretions, which the patient surely would have noted. The key here is that the blister ulcer formed would be painful and would not have the white fibrous coating, etc., noted in the clinical inspection. It's noted in the book that GWD, guinea worm dracunculiasis, may seem like some dermatological condition, seeing as this is This Week in Parasitism, not This Week in Dermatological Conditions. We can discount this. My friend pointed out cutaneous leishmaniasis, which seemed to fit the bill much more closely. Firstly, the non-tender lesion with a white fibrous coating is key to the diagnosis. It's the only non-tender ulcer noted in the book. The timescale also matches better to CL than to GWD, as GWD takes over a year to present with an ulcer, while CL, cutaneous leishmaniasis, would present within a month, as the patient said. Leishmaniasis is also common in South America. Costa Rica is noted for tourists especially. And cutaneous leishmaniasis does not often present on the foot, but as was noted in the notes, if the patient wore sandals extensively and was bitten there, it's possible that CL could show up there. Ultimately, the deciding factor for me was that Dr. Despommier seemed to get the diagnosis straight away. I guess that meant it must have been easy. I don't know. Problem solved. I've really enjoyed the show so far, listening to the backlog of episodes as I work. All the best, Luke.
Well, I'm not sure that him getting it quickly is a good indicator. Sometimes he'll be right, but not always, so be careful.
True.
All right, so the next 3 emails from Steve, Maureen, and Ted all have to do with the same case. They send a link to a ProMed article, CNN, and a Syracuse.com article. And this is, I'll read the ProMed mail, is cattle eye helminth, Thalasia gulosa in Oregon. First report, an Oregon woman has become the first person worldwide known to have an eye infestation by a tiny worm species previously seen only in cattle. that is spread by flies that feed on eyeball lubrication. 14 worms, less than half an inch long, were extracted from her eye over a 20-day period. So this is a worm only found in cattle. Wow, it's first ever. There you go. And so therefore Daniel has never seen it. And since Daniel doesn't treat cattle, you probably don't know much about this, but I do know that flies, they do settle on the eyes of animals like cows because they get fluids from them, right?
Yep. Now I think we had a case of the ovis areus, which is similar. It's like this, but it's in sheep. We had that surfer. A woman was surfing in northern Africa, and she actually got it in her eye because of the exposure to the sheep.
I just wonder how long it would take for the fly to light on your eyeball, and then I don't know how the worms come out of the fly, and so forth. It seems to me it would take some time, and maybe she was sleeping and it settled on her for minutes or something like that. But, if a fly goes on your eye and you're awake, you know it. You get rid of it. Right?
You would, yeah. And I think that's probably what happened. She probably was sleeping, right? Yeah. And then the fly landed, and that was how they got in there. Huh. Yuck.
Yeah, yep. Yep.
No, I remember when this case was reported.
All right, why don't you take Eric's?
Eric writes, hello again. From the description of a chronic painless ulcer with a raised edge originating from papule, I think our patient has cutaneous leishmaniasis. Before hearing of a trip to Costa Rica, I first thought of diabetic foot ulcers. However, they usually are purulent on the bottom of the foot and late complications of diabetes. Actually, the first thought that fitted through my imagination was Buruli ulcer, because it's a neglected tropical disease. Buruli ulcer is caused by a prokaryote, Mycobacterium ulcerans, and is endemic in Central Africa, not Costa Rica. I searched PubMed to help me guess which of the 15 New World species listed in your textbook were found in Costa Rica, specifically. In this review by a familiar author, I found that L. panamensis, brasiliensis, and infantum can be found west of Panama, with L. panamensis being the most commonly seen in Panama. I also found some similar case reports from Costa Rica that were L. panamensis. In these reports, PCR was used to identify the species. Don't know if the species identity was needed for this case, but I'm interested to find out, and he then gives us a reference. What I should comment that when we make the distinction between a Buruli ulcer, a Leishmania ulcer, or a bacterial ulcer, is a little bit of what was mentioned here. With the cutaneous leishmaniasis, you have a raised, indurated, so firm border all the way around, but it's not undermined, and the base of the ulcer actually looks clean, which is sort of amazing. Here we are in the tropics. How is this not super infected? The Buruli ulcer is really a disgusting ulcer. It is undermined. So if you take a Q-tip and you go right to the edge, you can actually go under the skin about an inch all the way around. So it's really like a very unpleasant ulcer. And your infectious ulcers, actually a patient recently where I was, going through, are you sure you didn't travel? Because this looks so much like a leishmaniasis ulcer, but they tend to be smaller. There's a different chronicity, tend to be more subacute, so you can't always tell right up front what it is. And there usually is a purulent drainage.
All right. The next few are also guesses. I'm going to go through these somewhat rapidly. Lana writes, Dear Leishmaniacs, I'm a longtime fan, but only an armchair biologist. Therefore, this reply is not intended to be a substitute for professional medical advice, diagnosis, or treatment. I want a signed book, though, so here goes. And this is the same case, Costa Rican with cutaneous leishmaniasis.
Yes.
All right. And then, okay, thank you. If you want to see these letters, by the way, they're all published on our website. Alan writes, Dear greetings, Professors TWiP. My guess for the patient with the lingering painless ulcer is leishmaniasis. Have been teaching in southeastern Nepal for 3 weeks and introducing medical students to your excellent online PD6. And was just talking with a local physician about the low but persistent prevalence of cutaneous leishmaniasis both in Nepal and in the refugees here from Bhutan. Your podcast seemed like a likely reminder that this infection is still around places just beyond just Afghanistan. Hope this gets in before your next segment. Keep up the great work. Alan is in Hawaii.
Yes, I remember Alan, University of Nations Global Health. So,
take the next one from Anthony, please.
Anthony writes, the black rat, Rattus rattus, originated in India but spread throughout Europe. The fleas that live on the black rat are thought to have transmitted the plague to people in Europe during the Middle Ages. The brown rat, Rattus norvegicus, came from Asia and generally replaced the black rat in Europe. The observation speculation is that the flea of the brown rat is species specific, and this limits the spread of plague. The black rat came to the west coast of the U.S. on ships from China. The black rat is thought to have brought the plague to San Francisco in 1900. And then he gives us a link to an interesting article on plague in San Francisco, though not focusing on rats.
I guess, we did talk about rats at one point recently.
Yes. Oh, yeah. And also fleas, right? We talked about Rothschild and how she showed how fleas jumped, jumping. My daughter Eloise did a whole school project, a whole write-up on plague in San Francisco, and the experiments trying to demonstrate to people there that the something on the rats was transmitting the plague. So
yeah, cool.
It was interesting stuff.
Whitney writes, Dear TWiP Trio, I found this paper today published in Scientific Reports by a group of researchers from the University of Notre Dame about how dominance rank of animals might affect parasite risk. I thought this might be an interesting paper to discuss on your podcast, as I also study parasites in a vertebrate animal system and have recently become interested in how parasitic infection connects to animal behavior. Presently 54 Fahrenheit in College Station, Texas, and partly cloudy. Here's Whitney. That's where Texas A&M is, isn't it?
I do not know. You have me there.
We are going there. Anyway, this paper, Social Status and Parasitism in Male and Female Vertebrates, a meta-analysis. They say, contrary to the prevailing paradigm that low status is linked to poor health, we found that dominant animals typically faced higher parasite risk than subordinates. So it's a study of many different studies.
I really like this article. I think I actually suggested to Dixon at one point that maybe we would want to discuss it, and was vetoed.
It was,
but it's a fun article actually. It's I think one of the problems with it, it's a meta-analysis, right? And meta-analysis is we can take all kinds of other analysis and you put them in a huge pile and you expect it to turn into something magical. And you really need to look at all the different articles, different studies individually to figure out their strengths and weaknesses. So there's a bit of an issue with meta-analysis, but it is interesting because a lot of these studies are finding that it's a little bit of a problem, and the high status males tend to have higher parasite burdens. And this is, I would say, a lot of studies are showing this.
Interesting.
Very interesting stuff.
Unfortunately, Dixon vetoed it.
Yeah, he vetoed it. It's I'm not sure. Maybe there was something that was even better, but it's a great study. So people that want to read about this.
All right. Let's do a couple more. Take Steve's.
Steve writes, Dear Twipsters, just a quick heads up on Nature's Citable,
yeah,
which I've not seen before. Here with some nice graphics is the story of the discovery of the apicoplast. And so what is this?
It is an educational venture by Nature Publishing Group.
Okay.
Where they have articles on basic stuff, not research articles, but they're for education. So this one's about the apicoplast, right? And it is an article that you could read to learn about it, and you could use it for teaching, so forth. So this one is really nice. Discovery, what it is, its past, definition. And you can find this for many different subject areas. There's a whole library of stuff. They started this not too long ago.
Oh, I like this. This is great stuff to share.
Yeah.
And this is All the Best from Stephen Hawkings.
Stephen Hawkins. Yeah, not the Stephen Hawkings, because the astrophysicist is, of course, no longer with us. This is Steve Hawkins.
Steve Hawkins as opposed to Steve Hawkings. But yeah.
Where it is trying to be winter again in England. Okay.
Okay.
Anthony writes, perhaps this rates a mention And he sends a link to a Stanford website called Parasites and Pestilence. So there's a list of parasites and then different years with links to descriptions of these parasites. I suppose like I just clicked on one and brought me to a web page. Yeah, it's basically a collection of parasites of different sorts over the years.
Yes, Stanford has a bunch of these parasite web pages that are pretty interesting. So, Rich writes, excuse the terseness, but just saw this on the MIT news page and wanted to be sure you'd seen it. Seems like a truly monumental advance in study and hopefully eradication of malaria. And let's see what he gives us a link to here. And it's human malaria parasites grown for the first time in dormant form. Okay, and this is an MIT news.
Can you explain what a dormant form is?
Let me see what they're trying to say here. So when I think this is sort of an issue with a lot of say pathogens is when things are in a sort of an active replicating phase, they tend to be vulnerable to a lot of our medicines. But if there's a dormant form of the parasite, then it's much harder to target it because there's a limited amount of dynamic activity. There's usually some dynamic activity going on.
This is a hypnozoite.
Yeah, so the hypnozoite is the classic dormant phase of malaria. It was actually described by an American, 70s or 80s, so it took a very long time. Vivax and Ovale have this, and so you could say the dormant form, the hypnozoite, is hypnotized. It's in this dormant, quiet state. And we're very limited in drugs that we can even use to treat this. They came out with a new drug recently, but then it was pulled because of toxicity. So when a person has, let's say, Vivax or Ovale, if you don't target and destroy this stage, they can, every so many months, actually have recurrences.
Cool.
So primaquine is really the only effective, safe drug that we currently have to target this stage.
So being able to culture these allows you to look for drugs that might target them,
which we could use.
Yep.
Definitely need that.
He says the weather in Boston is too damn. Why don't you keep reading that? It's got more there.
The weather here in Boston is still too darn cold. I changed the word from another word. Thanks for all your hard work getting to make science more accessible to the rest of us humans. Even computer geeks like me can understand at least some of what y'all talk about. Your shows are fun, engaging, and I think I'm actually learning something about biology. Take care all, Rich.
Nice. Lindsay writes, Dear Triosites, I'm a mechanical engineer who has used the back catalog of TWiP for several road trips. I'm also a regular listener to TWiVO and TWIM. Fortunately, I know some microbiologists and entomologists that I have put onto TWiP. I suspect that I am the only mechanical engineer with a copy of Parasitic Diseases 6th edition on my phone. The only case study that I have been able to get was when Daniel snuck in the recluse case. And this is only from having watched a bite point grow on the inside of my left elbow. The only problem that I have had in discussing the TWiP podcast is that my wife has forbidden it during meal times. I'm not exactly sure why. With the book, I'll start more serious attempts on the case studies. Lindsay is from Texas A&M University, where it's 80 degrees. 2 weeks ago in Big Bend, had 3 days bumping 90. Now, this email is quite old, so it's probably even hotter there.
David writes, Dear and esteemed parasitism panelists, it's Wednesday evening and I suspect the case is due for tomorrow. Initially, I thought the sick boy with the distended belly had Ascaris lumbricoides, but did not seem to explain the pale character, low weight, lack of responsiveness. Jumping ahead. In the end, although I'm probably wrong, I would guess it's a co-infection of both Ascaris and hookworm. Both are common, associated with dirt floors. Do you remember which case this was? David is writing to us from Hino Tepe.
It's one of your Panamanians.
Okay.
The boy, the Panamanian boy with the, I don't remember if he had
was
diarrhea.
Yeah, was this a heavy? It may have been a heavy ascaris infection, if I remember. Because I'm just thinking of a case. If you have distended belly, and hookworm infections usually are not very symptomatic.
Yeah, let me look it up here because you had 3 from Panama, right?
Mm-hmm.
So the farmer with diarrhea was the last one. Then before that was the woman with steatorrhea, right? She was Panamanian. And then
we had the boys with scratching themselves, right?
The itchy child from Panama, right? And then before that was the man with a leg ulcer from Panama. And then before that was the New Yorker with a rash. And we get these. Here we go. The 148, the South American child with belly pain.
Okay.
Do you remember that one? That was a while ago. I'll tell you the. So 148 was this 3-year-old boy, tropical South America, brought in by mother, says he's been sick for about a month, belly pain, poor appetite, constipated, goat stool pellets.
Oh, I think this was the strongyloides case, right? This may have been the gentleman. Yeah. Yeah, the really young one. That sounds to me like the strongyloides case, unfortunately.
All right, let's stop for now. Because it's 6 o'clock, and we have a bunch more, but we'll try and get those next time. And we'll try and tell you what cases they are if you'd like to know, because we haven't done a good job of that today. But it's been months since we did email, so these are ancient.
It's probably good for us to catch up a little bit.
We have to catch up next time as well. All right, we need a new case, Daniel.
Yeah, I almost forgot that. I was ready to sign off.
Yeah, you're right. We need a new case, so let's go back. I should have done that. I don't remember. We haven't done email in so long. I don't remember what the order is.
That is okay. Well, this is an exciting case, and I want to tell people this is a case that will be followed in about 2 weeks or so. We have a time when a colleague of mine, and this is a great story I like. This is a gentleman who I will say I was his mentor when he was doing his infectious disease training. He's now an attending at Columbia University in the infectious disease department, and he will be joining us in 2 weeks to do the unveil of this case.
He hasn't been on before, right?
No, this is a new one.
Okay, because you had someone on before that was here as well, right?
True. We had one of the pediatric infectious disease fellows, and before that I think we had one of the adult fellows, and so this will be
cool.
Our first Columbia attending, other than me, I guess, faculty presenting a case, or doing an unveil. So this gentleman, who was 79 years old, had a history of chronic lymphocytic leukemia, which had undergone a transformation into a diffuse large B-cell lymphoma. Because of this, he had received chemotherapy about 2.5 weeks before he initially was seen. When he was discharged with prednisone, and the prescription and the directions were prednisone 100 milligrams for 3 more days as part of the chemotherapy. But there was a little bit of confusion, and the pharmacy dispensed enough prednisone for him to take 100 milligrams every day for the next 2.5 weeks until he was finally seen by his hematologist. So he's taken a lot of prednisone. How
many weeks was that again?
Over 2 weeks.
Wow.
Yep. So over 2 weeks of 100 milligrams a day. The hematologist catches this, but the patient then goes on to have fevers up to 103 degrees at home. He reports a headache for which he's taking Tylenol. He also starts to develop a dry, non-productive cough. He didn't mention any of these symptoms to his hematologist, but a couple days later, these are still going on, and he is brought to the emergency room by his family. When he's in the emergency room, he reports that he also had not mentioned, but he has noticed he's lost a significant amount of weight, but he's not sure exactly how much it is, but he feels like it's notable. He doesn't have any nausea. He has no vomiting. There's no abdominal pain or diarrhea at this point. There's concern, and so they admit him to the hospital, and they continue the workup. When they see him in the emergency room, he is running a fever. They do a chest X-ray. And they see increased area of opacification, which they read as concerning for pneumonia. And he has started on broad spectrum, we'll say community-acquired pneumonia antibiotics, so ceftriaxone and azithromycin. But his fevers and symptoms continue, and at this point, they stop the steroids. They go ahead and they do a chest CT, and this shows multifocal ground glass opacifications and nodules. So they start to become concerned for some sort of an atypical or an opportunistic infection. They increase his antibiotics to a more broad spectrum to something called Zosyn, and at this point, infectious disease is consulted. We get a little more history at this point. He has a positive quantiferon gold test. This is a blood test for latent tuberculosis. But he says that he had already taken a full course of INH, so latent tuberculosis therapy. Also has a history of hypertension, coronary artery disease, an enlarged prostate. He's on a few medicines. He's on some prophylaxis because he's on chemo. So he gets something called Bactrim, which is a sulfur antibiotic, fluconazole, which is an antifungal, Valtrex, which is an antiviral. And he's also on amlodipine, Flomax. This is stuff for his prostate and blood pressure. We get a little more of that, what I call social or geographical history. He was born in the Dominican Republic, but he's been living in the U.S. since the 1970s. So he's been in this country for almost 50 years. He does occasionally visit, and he had been there a few months before. He's presenting. This is a recent case. We're seeing him in April, and he had been down in the DR in February for a short visit. He lives with his wife. He has an adult son. They're in the Columbian neighborhood, so the Washington Heights neighborhood. No pets, no smoking. Doesn't have anything he tells us about that runs in the family, and so no toxic habits, right? No smoking, no drinking. When he's initially seen, he's running a fever, Celsius 38.6. It's about 101 or so in Fahrenheit. His heart rate's in the 90s. He's requiring a little bit of oxygen. He is looking fatigued. He's coughing during the exam. He has scattered crackles in his lungs. He has a systolic murmur that he reports is old. The rest of the exam, his belly's soft. It's non-tender. He doesn't have any rashes. He's a little confused, but the family says he's always a little bit confused. His white count is 17,000, normal's up to 10. He only has 1% eosinophils. Platelets normal, mild anemia. Everything else is unremarkable. We're going to get a little bit more, and then you're going to have to try to figure this out. They're concerned, and so they put him on airborne isolation because they say maybe this is tuberculosis, maybe he didn't take his medicines. But they quickly rule him out. No positive sputums, and so he's taken off isolation. They say, oh, he's been on all the steroids. He's from the DR. We worry about strongyloides, right? So they send an antibody. That comes back negative. But he continues to spike fevers. And he has a little bit of improvement in his symptoms, but he's still requiring oxygen despite antibiotics. And they go ahead and they suggest a bronchoscopy, but he doesn't want that. And at this point, they do another white blood cell count. They're doing them every day. And at this point, his white count is increased. It's up to 25,000, and he has 30% eosinophils. So greater than 7,000, not greater than 9, but greater than 7,000. And so they are concerned now, oh, maybe this is a reaction to the Zosyn. We should stop that. So they stop that. The eosinophils continue to be elevated. They then start thinking of parasites. So they order a stool ovum parasite, but now he becomes constipated. So he can't produce any feces. So he has no bowel movements for several days, but now he's starting to have pain. So they pass a nasogastric tube to decompress him. He's starting to get a little confused. They CAT scan his belly, and this shows thickening of the colon, and they're concerned that it looks like something may be infiltrating. And so they go ahead and they do upper endoscopy, and they're going to do a biopsy of his intestines. And I will, as a hint, I will say we're going to get a diagnosis from that biopsy, which will then even confirm with some other testing.
So they're going to go from the top to get that biopsy?
So this is, they're actually going to do yep.
It's interesting.
Yep.
And that's because you don't have to do any prep for that?
It'll be interesting. This will be one of the things we can throw at our, it's going to be Justin Aaron who will be telling us.
Cool. It's a complicated case.
It is very complicated. So
he basically, his original prednisone prescription was filled wrong by the pharmacy, so he kept taking it when he should have stopped after a certain amount of time, right?
Yes.
Okay. All right. Very interesting. Maybe Dixon will be here next time. He'll get it like right there, I'll have to guess.
It'll be, what we'll have to do is before we let Dr. Aaron present, we'll have to get a little bit of feedback from what Dixon wants to know, if he's got some other questions or some thoughts. Because this is not a straightforward case, so I think it's a good case.
Not at all. I think we are scheduled at the moment for the 12th of July.
Yes. Just
to give people a little heads up, we will solve this case on the 12th. All right, that's TWiP. You can find it at Apple Podcasts, microbe.tv/TWiP. And now Google just started Google Podcasts, so that means if you have an Android device, you can play There's a podcast player now, and we have links to that in our webpage, microbe.tv/TWiP, on the latest episode, so you could just push that button on your phone and subscribe. If you like what we do, consider supporting us financially. We do need some help with our expenses, and we do shows on the road. We're going to Texas tomorrow. We're going to do number 500 at University of Texas, Austin. And then before that, though, on Thursday, we're doing an episode at Texas A&M. And I mentioned that because a couple of people noted that they are there. You can go to microbe.tv/tour for a list of all of our upcoming road shows. If you have questions, if you have guesses for the cases, TWiP at microbe.tv. I should say that if you want to give us some money, microbe.tv/contribute. I forgot to give you the link, microbe.tv/contribute. Questions and comments, TWiP at microbe.tv. Daniel Griffin is at Columbia University Medical Center and parasiteswithoutborders.com. Thanks, Daniel.
It's a pleasure, as always.
I'm Vincent Racaniello. You can find me at virology.ws. I want to thank Ronald Jenkins for his music. He's at ronaldjenkins.com. And ASM, the American Society for Microbiology, for their support, asm.org. You've been listening to This Week in Parasitism. Thanks for joining us. We'll be back soon. Another TWiP.
TWiP is
parasitic.